Good afternoon, everyone. I'm Dr. Philip Awadala, the National Scientific Director of CanPath, the Canadian Partnership of Tomorrow's Health. And I'm also the Executive Scientific Director of the Ontario Health Study, which is situated and hosted at the Ontario Institute for Cancer Research. So very excited about this webinar today. Welcome and thank you for joining. We're very proud of this particular set of series of webinars where we get to promote and highlight research from academics, in particular from research students, PhD students, master's students, and so on, who are using the CanPath resource. And so today we're highlighting two particular research scientists who I feel are probably going to be two of Canada's leaders in research in Canada's and cancer research in particular. So a few housekeeping items for today's presentation. If you have any questions for us, please use the Q&A button at the bottom of the Zoom window. We will be doing question and answer after each presentation, so the presenters will have about 20 minutes to present, and there will be 5 minutes for Q&A afterwards. If we don't have time to get to answer your question today, please reach out to CanPath at info@canpath.ca. And for researchers interested in data access, please contact access@canpath.ca. We also invite you to connect with us on Twitter, LinkedIn, and Facebook. I'd like to proceed with the land acknowledgement. We acknowledge the traditional territories of the Mississauga of the New Credit First Nation, Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous peoples on which the CanPath National Coordinating Center at the University of Toronto now stands. The territory was the subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lakes. With CanPath, teams working across Canada from coast to coast, we also acknowledge the ancestral territories that are home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this island. So our first presentation today is coming from Nicholas Cheng, one of the doctoral students in the Awadala lab. His research uses biological samples largely from the Ontario Health Study participants. Nick today will demonstrate how researchers can use blood signatures to detect breast, prostate, and pancreatic cancers about 7 years prior to clinical detection. And our second presentation is from Kimberly Skeed, another doctoral student in my lab. She's also the National Scientific Director, Coordinator for CanPath, and Kim will present how the interacting evolutionary pressures acting on somatic mutations in blood can be used to predict progression to blood cancer in large population cohorts like CanPath. So really quickly, we're going to do a quick introduction of Nick. Nick came to my lab from Imperial about 5 or 6 years ago now, and his PhD work is a great exemplar. Both projects are a great exemplar, but Nick's project in particular is working on solid tumors, and it's a great exemplar of how population cohorts can enable early cancer research and detection development. So Nick, I'm going to turn the presentation over to you.
Thanks, Philip. Is my screen seeable?
It is, yes. Thanks, Nick.
Okay.
Yep.
Okay, so thank you to the organizers for inviting me to speak today. I'm very excited to share some of the work I've been doing, particularly leveraging CanPath biologics to identify early cancer signatures. So I'll just start by giving a brief background in terms of the motivation behind using the specific biomarker we're looking at and why we're particularly interested in early cancer detection. So throughout our lifetimes, the tissues in our body accumulate genetic and epigenetic alterations. And while the majority of these mutations or changes in the epigenome are largely benign or non-consequential, a subset of them can potentially affect genes or regulatory elements. And in the context of cancer, some of these changes can be tumor promoting and can be broadly categorized into various cancer hallmarks. The acquisition as well as selection of tumor promoting driver alterations in both the genome and epigenome in cancers can be thought of as an evolutionary process in which subsequent accumulation and selection of these alterations can eventually give rise to cancers. If we look specifically at one of these alterations, specifically with DNA methylation, if you compare cancer normal genomes, two pertinent patterns can be observed. So, firstly is global hypomethylation. This is a decrease in methylation if you compare cancer versus normal tissue, and these tend to occur at repetitive elements of the genome. The second of which is local or focal hypermethylation, and these tend to occur at CpG dense regions. But instead of looking at individual individual regions, we can also look at multiple different regions across multiple cancers as well as normal tissues. So here, what we can essentially see here is that with DNA methylation patterns, we can identify regions that are lowly methylated in normal tissues, as you can see here, whereas they are hypermethylated in various multiple different cancer types. But additionally, you can also see or appreciate that within each of these cancer as well as tissue types, they all seem to have their own unique DNA methylation footprints or DNA methylation patterns. So from these two types of information, we can potentially use them for biomarker strategies. And just to simply demonstrate this, here we use TCGA methylation array data, where essentially we developed a predictive model using only from cancer and normal tissue from over 15 different cancer types. And this is from TCGA. And we essentially developed a predictive classifier that can inform whether a tissue type is cancer or normal just based on their methylation signature across 500 CpG sites. And you can appreciate here the extremely high sensitivity as well as specificity for classifying whether a tissue is cancer or normal just based on their methylation signatures. Likewise, on the flip side, instead of looking at whether or not a tissue is cancer or normal, we can also potentially inform the tissue type of a cancer, again, using their methylation profiles. So again, here we also use TCGA data. And what we've done here is we've plotted the true class on the left over here, as well as the predicted class predicted by the tissue of origin classifier on the bottom here. And you can appreciate by the color across the diagonal in this confusion matrix that we can identify with remarkably high accuracy the tissue of origin of a cancer. So we can potentially distinguish between a breast cancer from a kidney or stomach cancer using just their DNA methylation signature. So we can potentially use these two axes of information, both whether or not a tissue is cancer or normal, as well as the tissue of origin of a cancer based on their methylation signatures, particularly for early cancer detection. And this is particularly promising because if we look at population-based screening recommendations for cancer, they're currently limited to a few different cancer types, namely breast, colorectal, as well as cervical cancer in the healthy population, as well as low-dose CT for lung cancer screening among smokers, which means that for the majority of other cancers, they're typically diagnosed when someone presents a visible lump on the surface of the skin, or when they become symptomatic and go to a clinician for follow-up diagnostic. But if we look across survival rate across cancers, you can appreciate here that the earlier the cancer is detected, particularly when it's still localized to the tissue of origin at the time of diagnosis, that's when the survival is extremely high. You can see across virtually all cancers. But the problem is when it's diagnosed when it's already spread to more regional or metastasized to more distal parts of the body, and that's when survival rates are significantly lower. So if we could potentially shift the window of detection from these among these participants that are often diagnosed at regional or distal stages towards these localized stages when survival is the highest, we can potentially have extremely high clinical impact to improve cancer survival rates. So one idea here is to take advantage of the fact that when cells die in your body, they shed DNA that is freely circulating in the blood, and this is called cell-free DNA. And because this is seen in both healthy as well as cancer tissues, we can take advantage of the fact that cancer-derived DNA tends to carry unique genetic as well as epigenetic markers. So the idea here is if we can potentially identify whether or not a cell-free DNA fragment is from a tumor just based on their methylation signatures, we can potentially inform whether or not someone has an underlying cancer just from a single blood test. And indeed, it's been well demonstrated across multiple studies to date that if you profile a blood sample from someone that is diagnosed with cancer, and you try to look for tumor-specific cell-free DNA markers, either through mutation or DNA methylation profiling, you can indeed capture concordant markers that are from the tumor themselves. The problem with a lot of these studies to date is they tend to profile from individuals after they're diagnosed with cancer. And this tends to bias patient recruitment towards individuals that are already diagnosed with a late stage or are already symptomatic with the cancer. So what we would really want to do to infer whether or not these predictive markers are actually working in an asymptomatic individual would be to collect these blood samples before someone would even be diagnosed with a cancer. And one solution to this is to leverage large population cohorts such as CanPath, where they collect biologics from participants when they're first enrolled to the study. And in this work, we specifically focus on biologics collected from the Ontario Health Study. So the idea here is at the time of study recruitment, a large proportion of these participants will have provided both their health and lifestyle information through questionnaires, and they'll also have had their biologics collected when they're first enrolled. And this is primarily through blood samples that are later stored in the biobank. And over time, as we follow up with this cohort, as they continue to age, and over time, a large majority of them will be healthy when they're first recruited to the study, but later on will be diagnosed with a cancer. And these are called our incident cancer cases. So the idea here is we follow up with these participants who donated a biologic and were cancer-free at the time of study recruitments, but were later diagnosed with either breast, pancreatic, or prostate cancer. And in this work, we specifically focused on breast and prostate cancer because they're the two most common types of female and male cancers, respectively, which means that we had higher incident rates for these cancers, allowing us to collect more biologics. On the flip side, we focused on pancreatic cancer because it tends to be one of the cancers with the most poor prognostic rates. This can be partially due to late stage diagnosis of the disease. So the idea here is we identify participants that developed a breast, pancreatic, or prostate cancer after they were enrolled into the study. And just to give you an idea of the time between sample collection and diagnosis, here we plotted the distributions, and they're colored by the stage in which they were diagnosed. So you can appreciate here that we have some samples collected relatively close to diagnosis within, say, 1 or 2 years. We have some blood samples collected over 4, 5, or even 6 years before they were diagnosed with the cancer. And likewise for prostate and pancreatic cancer, you can see here. You'll also notice for breast and prostate cancers, they're typically already diagnosed at a relatively early stage 1 and 2, whereas for pancreatic cancers, they're often diagnosed at stages 2, 3, or even 4. So in addition to each of these pre-diagnosis blood samples, we also selected plasma samples from cancer-free controls, and they're matched to each individual case by age, sex, when the sample was collected, as well as other health and lifestyle factors. And the idea here is from each of these pre-diagnosis cancer cases, as well as controls, we take 1.8 mils of the plasma that is stored in the biobank, and we can profile their cell-free DNA methylation signatures using an approach called CF-MediPseq. And the gist of it is we use monoclonal antibodies to bind to methylated cell-free DNA fragments after we isolate them, and we pull them down and sequence them to infer which regions of the genome in cell-free DNA is methylated within each of these pre-diagnosis cancers, as well as cancer-free controls. So what we try to do after we computed the counts across non-overlapping tile regions of the genome and we normalize the counts, what we try to do is we try to first see, can we identify any discriminatory signatures in their cell-free DNA methylomes between our pre-diagnosis cases and cancer cases and cancer-free controls. So what we did here is so first here with breast cancers, we identified 250 regions that are the most hypermethylated in our pre-diagnosis breast cancer. So these are regions that have a higher level of methylation or are uniquely methylated in our pre-diagnosis breast cancers. And we can visualize how well they discriminate between our pre-diagnosis cancer cases and controls using an approach called principal component analysis. And the way you can infer this is that the closer the dots are together, the more similar their signatures are. So you can see here that we can not only discriminate between our cancer-free controls in these square gray boxes here from some of the pre-diagnosis cases collected 1, 2, or 3 years prior to diagnosis, such as those in blue and green. We can also separate some of the samples collected 3, 4, or even over 5 years prior to diagnosis, as labeled in red and yellow. And likewise, if we look across prostate cancer, where we've profiled a little over half of our samples so far, as well as our pancreatic samples, we can start to see these discriminatory signatures in our cell-free DNA methylomes in samples collected even over 4 or 5 years prior to diagnosis. What we also try to do here is we also acquired cell-free DNA methylation profiles from samples collected at the time of diagnosis from a few different studies. And what we try to do here is we project using the pre-diagnosis cell-free DNA methylation signatures, we projected on samples collected at the time of diagnosis, as indicated by these purple triangles, and looked at where they would position in our principal components relative to the cancer-free controls and pre-diagnosis cases. Again, you can see along the first principal components that we can see the transition across the 3 cancer types from the cancer-free controls in gray to the pre-diagnosis samples all the way to the post-diagnosis samples. So you can see that the spread among the post-diagnosis is a lot higher. And we saw this as for all 3 cancers. So what we try to see next was to see among the features we capture, are they actually from the originating cancer? So are the features we're capturing in the cell-free DNA concordant with what we would observe to be methylated in the cancer tissue themselves? So unfortunately, while we didn't have the tumor of these individual patients, what we try to do instead is we try to compare the hypermethylated regions in cell-free DNA with tissue methylation signatures. So what we did here is we try to first compute the difference in methylation between breast cancer tissue from TCGA relative to peripheral blood leukocytes from data across the literature. So these are samples from another data sets. And what we try to do is we try to see among the regions that are significantly hypermethylated in breast cancer relative to blood cells, are they the same, or do they overlap with regions we're capturing in the cell-free DNA? And the overlapping count is what you see in these bar plots above right here. And we wanted to see how enriched this is compared to if we were to just randomly select regions of the genome and looking at whether they overlap. So the idea here is to see how enriched we are for regions that are hypermethylated in breast cancer tissue relative to blood among the signatures we're capturing in cell-free DNA. So these box plots indicate a random permutation, looking at random overlaps between random non-significant regions versus tissue methylation regions, and these red dots indicate the actual overlap we observe. So what you can see here is among the regions that map to CPG islands and shores, we're significantly enriched for regions that are hypermethylated in breast cancer, which makes sense because a lot of the regions that tend to be hypermethylated in cancers typically occur at these CPG-dense CPG islands or CPG shores as well. And what we also try to do is we try to see among the signatures we captured in the pre-diagnosis cell-free DNA, can we discriminate between tissue types based on their methylation signatures. And again, using this TSNE, you can see not only can we discriminate between peripheral blood leukocytes in this in the gray color here from these breast cancer and breast normal tissue in this clump here, we can also discriminate between breast normal from multiple other breast cancer subtypes. And you can also appreciate here, we're also able to discriminate between luminal breast cancer subtypes from basal subtypes using cell-free DNA pre-diagnosis signatures. So this essentially informed us that a lot of the features we're capturing are indeed originating from the breast cancer themselves. But what we really wanted to try and see is how predictive are these cell-free DNA methylation signatures for predicting whether or not someone has a cancer from these blood samples collected over 3, 4, or 5 years before diagnosis. So what we did here is we used a typical machine learning pipeline where we separated our samples into our training and test sets. So with our train set, we performed feature selection as well as DMR calling and built a predictive model. And we looked at how well that predictive model works by testing it out on the held out test sets. And we repeated this in essence over 2,000 times with random training and test splits such that we can infer the generalizability as well as the overall predictive performance for classifying these pre-diagnosis pre-diagnosis cancer samples. So here I'm just showing you the results for our pre-diagnosis breast cancers where we profiled all our samples. And this solid line in the middle here indicates the mean predictive performance across these 2,000 iterations, and the shaded regions indicate the 95% confidence intervals. And you can see from this AUC curve, we're getting around an average AUC performance of 0.77. We're capturing about almost 50% of our pre-diagnosis breast cancers just based on the methylation signatures, which means that we're performing particularly well. But more interestingly, when we looked at subgroups of our participants, so here what we did is we stratified our performance, just looking at cases for some of these particular subgroups, we noticed two interesting cases. The first here is that we had a subset of participants that tested negative for cancer through a mammogram screening within 1 year blood sample collection. So these are participants that went through a mammogram, tested negative, but also had these blood samples collected within a year of testing negative. And among these participants with a negative mammogram screening, we're capturing almost 50% of these participants with a positive cancer signature from their blood profile before they were even diagnosed with cancer. Secondly, we also noticed that among individuals diagnosed before the age of 50, so these are participants that have developed an early breast cancer before mammogram eligible screening ages in Ontario, we found that we're doing particularly well in capturing some of these participants. So here we've identified two groups that would typically be missed by a mammogram, but we were able to capture a potential cancer signature from their blood sample before they were even diagnosed with the cancer. What we also try to do is we try to see whether or not the pre-diagnosis signatures we capture among our pre-diagnosis cancer are generalizable to samples collected at the time of diagnosis. So what we try to do here is using the most informative features from these 2,000 repeats. We developed a final predictive classifier using only our pre-diagnosis samples and OHS samples, and we tested how well that classifier works on samples collected from other clinical studies as well as controls from other cohorts as well. So you can see here when we looked at when we validated our pre-diagnosis signatures and samples on these post-diagnosis samples, we're getting remarkably high diagnostic accuracy. We're capturing almost over 72% of these breast cancer cases collected at the time of diagnosis at both early as well as late stage cancers with remarkably high accuracy. And likewise for prostate as well as pancreatic cancers, again, you can see that for prostate cancers, we're also able to capture both early as well as late stage prostate cancers with relatively good predictive diagnostic accuracy. And for pancreatic cancers, all of these participants were diagnosed at metastatic stages. So what you can So what we're trying to do here at the moment is we're trying to look to see if we can capture some of these signatures in pancreatic cancers when they're still largely localized or in relatively early stages. So as you can see here, I hope that you can appreciate the amount of work as well as resources that large population cohorts build upon to enable to leverage emerging technologies with these resources to further our understanding towards early cancer detection using blood signatures. And this type of activity can be potentially applied to not only early cancer detection, but also other chronic conditions to understand their early evolution. Likewise, by leveraging these population cohorts, we can profile these pre-diagnosis and pre-diagnostic blood samples to identify how early we can capture some of these early cancer biomarkers. And in this case, with cell-free DNA methylation signatures, we can potentially capture some of these signatures up to 7 years before someone would typically be diagnosed with a cancer. And finally, we can again leverage these cell-free DNA methylation signatures. And while they've been demonstrated to be predictive of cancers at the time of diagnosis, here we're able to demonstrate that we're able to capture some of these signatures several years before someone would even be typically diagnosed. And with that, I'd just like to thank my supervisor, Philip, as well as the rest of our lab members, Elias, Tom, Kim, as well as Dave, as well as our collaborators at the Princess Margaret Cancer Center, particularly Scott Bradman, Daniel DiCavallo, Dave Sescon, as well as Hanson Hay, as well as the Genome Research Platform team at OICR, who helped perform the CF meetup-seq assay for our OHS blood samples. And with that, I'll take any questions. Thank you.
Great, thanks very much, Nick. That was a fantastic presentation. It's a great example of how biobanked material with proper linkages to, I think, administrative records can enable prospective as well as retrospective research. I'm going to turn over the question answer period to John McLaughlin, who's the Executive Director of CanPath. And John, I see you're on the screen, so you could take it away from here, and I'll do the introduction to Kim afterwards.
Thank you, Philip, and particularly thank you, Nick, for tremendous work, great summary and clear presentation. So there have been a number of questions posted in the Q&A, and I encourage everyone to add to that. And Nick, there are a couple of questions that I can turn your way.
Sure.
One from Esther is asking the question of essentially the timing of the biospecimens that you've been analyzing in your study. And she noted too that there may have been, as part of other CanPath work, additional samples collected as part of the COVID work. So Nick, for your paper today, can you comment on when those biospecimens that were banked were collected, and perhaps even whether there would be potential value in the future of looking at repeated biosamples, but I believe that you did not have an opportunity to look at repeated biosamples in your paper today. Is that correct?
Yes, that's correct. So the majority of these blood samples were collected between 2009 to 2017. And these are all single blood samples that we use. So we only use samples that were collected at the time of study recruitments. So at the moment, we're not using these blood samples for COVID work specifically, but I am aware that CanPath has other activities. So they did take blood spots, and they're planning to do additional COVID work with those samples. But in terms of the blood samples that we use for today's activity, they're mainly used for cancer research.
And then furthermore, the specific very small amount of biospecimen that was collected for the COVID work, it's largely been focused on the amount necessary to do antibody testing. Philip, was there anything else you wanted to add about the potential for a link between infectious disease research and COVID and cancer research, given the specimens collected by OHS?
Yeah, that's a great question, John. So actually, our next presenter, Kim, is going to be working on a project that's related to how some of those COVID specimens themselves can be utilized in this kind of work, largely in the context of how blood mutations develop over time and how that might be impacted either by pre-infection or potentially going forward, how they might impact things like response to vaccination as well. So no, I don't want to add too much more to what Nick just said, how Nick just answered that.
Thank you. Another question, Nick, relates to what are the implications of Chantel has asked the question, if a sample is found to have cancer cells in it, would patients get contacted and be informed? And given the design of your study where all of these were cancer patients versus controls, can you comment on whether there would...
So if these participants are indeed really high risk for having a cancer, then they would indeed be contacted. But a lot of this work is still in the testing phase. We're still actively developing it to improve the predictive performance. We don't want to overdiagnose patient or unnecessarily inform them if they potentially have a cancer, but they in fact don't. But it is something that if we are really positive that these individuals do have cancer, then we would potentially contact them. But at the moment, because a lot of the controls we found do not have a cancer signature, we haven't really had to do that, as well as a lot of the pre-diagnosis cancers we're capturing, they've already been diagnosed with a cancer. But in the example that you give where a control sample would test highly positive, then we would potentially inform them.
Thank you. Jonathan has asked, how would you define CPG islands?
So this is mainly using reference data sets out there. So here I use the UCSC data sets out there that have publicly annotated CPG island shores and shelves across the genome. If that's how you would define it. The main definition for CPG islands are particular regions of the genome that have a higher CPG density, and they tend to be at 60 or 70%, have the CPG motif on it. But in the data set I use reference data sets out there to inform which regions to annotate.
Thank you, Nick. Regine has asked for clarification on the sample size, which I believe one of your early studies showed there were 136 breast cases, 16 pancreas, 252 prostate, and 404 controls. I think,
right.
Is that correct? So
yes.
Regine, I think that would answer your question. It was in one of the earlier slides. And by the way, for all present participants, the slides will be available after today's presentation. And indeed, the webinars are recorded and available through the CanPath website. Stephen has asked, what type of predictive models did you run, e.g. logistic, machine learning, and if you ran multiple models, did performance differ by the model type? So the question was about predictive models.
Yeah, so here I did try a bunch of different predictive models. We tried SVMs, neural nets, random forest classifier, logistic regression classifiers, other penalized regression approaches. What we found was that random forest models seem to work the best, and then logistic regression classifiers or like elastic net models tend to perform almost as good. But those are the two that tend to work the best: random forest models as well as logistic regression.
Quite a few questions that have arrived here. Let's see, how long does it take to analyze these blood samples, and how expensive or labor intensive are they to run?
Yeah, so I guess one of the, so in terms of how long it takes for a blood sample to go from the biobank to generating the sequence data we want, it does take a while to get all the samples together, and then it takes around 2 weeks for the assay to be performed, the CF medium-seq assay. But the problem is that a lot of groups are trying to do CF medium-seq at the Genome Research Platform, so it tends to be a lot more hands-on in terms of how much work needs to be done to perform these type of profiling. So it does take a few months for these profiles to come in, for all of them to be sequenced, and for us to start analyzing them. And in terms of how expensive, At the moment, I think to perform the assay themselves, I think it's around somewhere around 200 to 400 Canadian dollars. I don't remember the cost off the top of my head. But if you're running a lot of them at the time, then you might be able to multiplex the samples a lot more. So it might be a lot faster. But if everything went smoothly, it wouldn't take more than a month, presumably.
Okay, thank you, Nick. In the interest of time and allowing for the other speaker, maybe I'll just ask one final question here. Let's see. Are there any examples of how this method could be applied to detect other types of chronic diseases besides cancer?
Yeah, for sure. So one of the things that's been noticed about cell-free DNA is whenever you have damage at a tissue sites, there tends to be more cell-free DNA shedding from that tissue of origin. So it's been demonstrated, for example, in people with organ transplants or cardiovascular conditions, that there tends to be increased shedding of cell-free DNA from the originating tissue sites. So one idea that we're currently working on, as well as is being done by a lot of groups, is to see whether or not we can deconvolute the proportion of cell-free DNA fragments from individual tissue sites, such that we can infer whether or not, say, if someone had increased in shedding from liver tissue based on their cell-free DNA profiles, we can infer that there could be some kind of chronic condition in that specific originating tissue. So it's been demonstrated in some autoimmune conditions. It works especially well for autoimmune diseases. But there's more and more work out there that's been showing this for various other conditions like cardiovascular diseases, potential neurological disorders, as well as some other conditions.
Okay, Nick, thank you again for a tremendous presentation, great answers. To participants, a few of the other questions we'll try to provide answers through the Q&A, through typed answers, and I'll turn it back to Philip, who can actually introduce the next speaker. So again, thank you.
Thanks very much, John. Again, thanks again, Nick, a great presentation. And John, we'll see you again in about 20 minutes or so. So great pleasure to present the next speaker, Kimberly Skeed. Kim came to my lab from Trinity College at University of Toronto some number of years ago now, and while she's been doing her PhD, if you've been working at all with CanPath, you may have come across Kim working with us, because she works at the National Coordinating Center as our working directly with many of us across Canada, supporting the NCC as our scientific coordinator, and so has been able to do a lot for CanPath, as well as do a lot for supporting a lot of the work that we just saw from Nick's project around cancer linkages. And that's really important work for CanPath as a resource as well. So I wanted to highlight that specifically. Stay tuned to what we're calling the Canadian Cancer Study, which Kim is leading. So Kim's doing very pertinent research as well with regard to early cancers, but working more on the blood side rather than what we just heard from Nick with regard to solid cancers, although the work that she's doing in blood does have, and you'll see today, some impact on solid cancers as well, and some predictive potential. So this is a particular area that Kim's working on. Is a very topical area, and it's a very exciting line of research, and so very happy that she's presenting to us today. So take it away, Kim.
Thanks, Philip. Just confirming that you can see my screen. Can you see my slide?
Yeah, we can see everything. We can see your title slide. Sure. Yep.
Wonderful. Great. So thanks so much for the introduction. I'm really excited to be chatting to you today about my work, which is focused on predicting health outcomes from hematopoietic evolution. So how does our blood change as we age and progress to disease? This idea of taking a more personalized approach to understanding health outcomes as we age is really in keeping with the larger global shift towards precision medicine. Typically, what this looks like is we take populations of people and we profile them biologically. So looking at things like DNA, blood samples, tumor samples, and using that information to start to stratify people into different treatment groups so that we can start to administer more tailored therapeutic interventions in a way that's likely to optimize that individual's chances of having a positive health outcome. What I'm particularly interested in is what is driving the onset of disease in the population in the first place. And there are a number of key players that we can think about here, such as the environment, genetics, behavior, and lifestyle, age, and how all these factors work together to shape the patterns of mutations that we accumulate over our lives. One way that we can understand those patterns of mutations is through the application of a population genetics lens. This is something that we're really comfortable doing at an individual level and, for example, studying tumor evolution, because tumors all start off as healthy populations of cells which start to pick up mutations as time goes by. The vast majority of those mutations we expect to be mildly damaging or completely neutral, term passenger mutations shown here in yellow. But every once in a while, we're going to get an event known as a driver mutation shown here in red, which pops up and confers some sort of growth advantage to those cells, causing them to expand in frequency and form that mass that we call a tumor. What we can do with population genetics is we can sample a tumor at a given time point, and then through studying the frequency of mutations in that sample, make inferences about what happened in the evolutionary history of that tumor. What we are doing is we're taking some of these very classic population genetic tools and using them to understand how our blood changes as we age. We can do this because the entire human hematopoietic hierarchy, our entire blood pool, is rooted in a small population of stem cells known as hematopoietic stem cells, which reside in our bone marrow. These cells are highly productive and are ultimately responsible for all the mature cells that we see in our blood. And naturally, because they're so highly productive, they need to be very tightly regulated in order to maintain the composition of the mature blood pool. However, something which people have been paying a lot of attention to lately is this notion that as we age, we start to accumulate somatic mutations either in our stem cells or in early progenitor cells, which confer a growth advantage to those cells, termed clones, causing them and all of their derivatives to become disproportionately represented in the mature blood pool. These mutations can be quite heterogeneous in form, ranging from single point mutations to large structural changes to our chromosomes, which have been detected in around 5% of individuals. But typically, when we see that one clone, so one cell and all of its derivatives, have become represented at a frequency of greater than 2% in our blood, we say that that individual has clonal hematopoiesis. And because this is a phenomenon which has been closely linked with age, it's been termed age-related clonal hematopoiesis, or ARCH. Something which is incredibly common in the adult population. We think that it's almost inevitable in the elderly, but the reason why people have been paying so much attention to it lately is because it's been found to associate with around a 10 to 14-fold increase in likelihood of later progressing to cancer or cardiovascular disease. And it's thought that in those individuals who progress to malignancy, they're accumulating somatic mutations in known cancer driver genes. There have been a number of genes such as DNMT3A, TET2, ASXL1, which I'm highlighting here, which tend to be recurrently mutated and are associated with later disease progression. But if we look at the proportion of individuals in the population who are healthy and harbor a mutation in a known driver gene, which I'm showing you here in gray, compared to the proportion of individuals who progress to disease, shown in red, those numbers look quite different, and there's some fairly massive implications to this. If we start responding to that gray group, we're going to end up hugely overtreating a huge chunk of the population that never really requires that therapeutic intervention in the first place. But if we don't respond to that red group, that group who subsequently progressed to disease, we're missing out on an incredibly valuable opportunity to capture cancer in its infancy when treatment outcomes are much more likely to be effective. What we are proposing is that the background on which these driving mutations accumulate matters. By that, I mean, we have a very clear idea of what positive selection looks like when it's acting to increase the frequency of a driving mutation in a population, causing somebody to shift towards that tumor phenotype. We also have a very good idea of what negative selection looks like when it's acting to remove those mildly damaging mutations from a population in order to maintain the integrity and the health of your tissues. What we don't quite understand is what happens when we get a positively selected driving mutation arising in the same cell that has already accumulated a number of mutations and a negative selection. We might start to see some competing evolutionary pressures that could be acting to inhibit those disease-associated clonal expansions. Of course, we also have to consider a scenario where blood evolves in an entirely neutral manner, and any rise and falls in clonal frequency occurs purely by chance. And here I'll just highlight that cancer is in fact most frequently studied through that lens of positive or neutral selection only. So this question really informs the overarching goal of our study, which is to investigate how interacting evolutionary pressures in our blood might be shaping health outcomes as we age. So first, we're interested in looking at how our blood evolves as we progress to disease. Are there different types of selection at play here? And second, if we think that selection is playing such a big role in shaping our blood and our subsequent health, why are some large structural mutations tolerated to such a great extent in our blood at all? And so to tackle this first question of how does our blood evolve as we progress to disease, we generated over 5 million artificial blood populations, each evolving under one of those 4 evolutionary models that I described earlier: positive, negative, neutral, and combination models of evolution. Each simulated population here represents an individual blood pool, so a blood sample, and can be collapsed down into a very concise set of summary statistics. So looking at things like the number of mutations in a population, the type of mutations in a population, and the frequency of mutations in a population. And what we can do then is we can take those summary statistics and use them to train a deep learning classifier that attempts to reverse predict that overarching evolutionary class that was used to simulate that blood population. When we apply our classifier to a held out test set of simulated data, we find that we're able to distinguish between the different evolutionary classes very well. We can predict the true evolutionary class correctly around 86% of the time, as you can see here in our confusion matrix. Where we start to fall short is in distinguishing between negative and neutral classes of evolution. But if we take a step back and think about what's going on biologically here, negative selection is removing mutations from a population, and our model is using mutations to make this prediction. So we're quite simply just reducing the amount of information that we have to work with. Regardless, this is a long-standing challenge of population genetics, distinguishing between these 2 different classes, and we were still able to get it right over 80% of the time. And because of this, we decided to move forward with applying our classifier to capture how blood evolves leading up to disease. To do this, we leveraged a collaboration with John, Dick, and Sagi Abelson at OICR, who had drawn from the European Perspective Investigation into Cancer and Nutrition Study, so a large population cohort in Europe, to identify around 100 individuals who provided a blood sample about 7 years prior to progressing to acute myeloid leukemia, but 400 age and sex-matched controls. All of those individuals underwent targeted sequencing at a very high coverage, around 5,000x per individual, and we use the sequencing data to generate those same input statistics that I described earlier so that we can classify the evolutionary dynamics in the blood of those individuals. Right away, we start to capture some pretty exciting results. And specifically, we find that the vast majority of individuals are rejecting those models of neutral and positive selection only, which are so frequently used to study cancer evolution. And we're finding that over 60% of individuals are showing signatures of negative selection, either acting alone or in combination with positive selection. And when we zoom in on individuals fitting this combination model a bit more, we find that we're able to capture a significantly higher ratio of passenger to driver mutations in individuals who remain healthy, which are shown here in blue, compared to those individuals which ultimately progress to disease, effectively suggesting that negative selection might be playing a role in keeping these individuals healthy, as they're quite simply more targets for it to act upon. So naturally, our next question was, well, where in the genome are these mutations occurring? And do we see enrichment of certain genes mutated in these different evolutionary classes? And what we find is that very few genes are exclusively mutated within a single evolutionary class, but that many genes are commonly mutated in those neutral, combination, and negative classes of evolution, which I'm showing you over here. As well, we find that a very small number of genes are exclusively mutated in negative neutral models and positive neutral models of evolution. But coming back to the overarching goal of our study, how do these different evolutionary models impact overall progression to disease? And to study this, we performed a survival analysis and found that individuals who are fitting those negative only or neutral classes of evolution are in fact remaining AML-free for significantly longer than those individuals with signatures of positive selection. As you can see here, we couldn't really distinguish between the combination of positive models in this instance because we had so few number of individuals who fit that positive only class of evolution. And if you're interested in hearing a little bit more about the story, I do encourage you to check out a paper which came out at the end of last year, which I've listed down here. But the key thing that I want you to be taking home here is that we think that selection acting on passenger mutations is in fact playing an important role in shaping clonal dynamics in our blood, and that we do need to start adopting a less driver-centric approach to fully understand cancer evolution and outcomes. This really leads us quite nicely into our second question of, given that we're seeing all of these different selective pressures in our blood, how is it possible that we see so many healthy individuals in the population who harbor large somatic structural variants, so large changes to our chromosomes with seemingly few consequences? Presumably, these would be selected out. And this is something which really captured our attention after a beautiful study came out of the UK Biobank in 2018, which found that up to 5% of the population, so the healthy population, harbor a large somatic structural variant. So typically a loss of DNA, a gain of DNA, or a loss of heterozygosity in their blood pool. Because we're so astonished at this high number, we set out to capture somatic structure variation in about 20 different populations, specifically the Canadian population cohort study, CanPath, which has brought us all here today, as well as the 1,000 genome study. And more specifically, through these cohort activities, we had access to a variety of different genotyping array data that we were able to use to identify these gains, losses, and loss of heterozygosity events across the genome for around 16,000 individuals. And I'll just mention that a key advantage of the CanPath code here is that we have access to much denser sequencing array data. So here I'm specifically referring to the Omni 2.5 array, and these arrays are much more sensitive than what have been previously used to study somatic structure variation, which really enabled us to capture an increased frequency of these events across the population. And so as you can see here, by looking at this yellow bar, we're finding that approximately 3 times as many individuals in the population, so around 15%, have at least one detectable structural variant in their blood compared to the previous estimates, which I'm showing you in the dashed red line. And further, quite remarkably, when we start to look at the relationship between the size of the structural variants and their frequency, shown here as cell fraction, so this means how many cells in your blood population are actually harboring these large structural changes, we start to see this quite lovely relationship emerging where small variants are able to rise in frequency in our blood and large events tend to segregate at incredibly low levels. And we think that this is indicating that selection might again be playing a role in shaping the clonal dynamics of somatic structural variability. So naturally, because we think selection is playing a role here, our next question was, well, where in the genome are these mutations occurring? This might tell us a little bit about the types of selection that might be acting on these variants. And as you can see here, where I'm showing you the distribution of gains, loss of heterozygosity, and loss events across the genomes, these are accumulating quite broadly and are largely quite rare events in that it's not uncommon for us to see a single structural variant covering one single region of the genome. But we wanted to find a way to better parameterize areas that are enriched for somatic structure variation. So we're calling these hotspot locations in the genome. And we did this by adopting a relatively simple approach where we just moved along the genome, querying whether each SNP was more impacted by somatic structural variation than expected by chance alone. And in deriving that neutral expectation, we considered a variety of things such as the type of structural variants, the chromosome on which they were occurring, and the sensitivity of the rays that were used to detect them. And what this lets us do is it lets us start to map out regions which are disproportionately impacted by structural variation in the genome. And here you can see those hotspots plotted across the chromosomes, with the colors indicating the structural variant type and the shapes indicating the array that were used to detect them. And everything above that dashed red line would be an area which is significantly enriched for somatic structural variation. When we zoom in on a couple of regions, so here I'm just showing you the hotspot locations on chromosomes 2 and 13, we start to see some of these really nice peaks emerging of structural variant enrichment. And when we look at where in the genome these peaks are, we find that they typically tend to accumulate over a number of genes which have been previously implicated in the development of age-related clonal hematopoiesis or blood cancer. And specifically here, I'll just highlight that we're seeing an enrichment of loss events at known haploinsufficient tumor suppressor genes, such as ASXL2 and DLU1, 2, and 7, meaning that this loss might be a first step in catalyzing that shift over to later cancer progression. Supporting this theory is the fact that these hotspot locations across the genome are conferring a clonal advantage to cells harboring those mutations. As you can see here, when we compare the cell fraction of the structural variants which overlap with a hotspot region shown here in red, compared to those structural variants which do not overlap with any hotspots shown in gray, the ones which are overlapping with a hotspot are seeming to be more successful and start to take over our blood pools. So, of course, the natural next question is, well, are these individuals progressing to blood cancer? And to figure out if we are seeing an increased rate of cancer progression amongst individuals who are harboring these somatic structure variants, I linked one of the regional arms, the Ontario arm of the CanPath cohort, to administrative data collected by Cancer Care Ontario. So you would have heard Nick chatting about this a little bit earlier. Cancer Care Ontario is a data provider we have here in Canada. And these types of linkages really enable us to capture clinical outcomes for all participants in the cohort, including cancer outcomes at the ICD code level. So because these types of linkages are quite challenging for us to do in Canada, we initially only piloted this activity for blood, breast, prostate, and pancreatic cancer. And what we find is that in our cohort of individuals who've been genotyped in the Ontario Health Study, we're able to identify around 109 individuals in the cohort who provided a blood sample prior to progressing to malignancy, and that the presence of at least one somatic structural variant in that group tends to be associated with around a 3.5-fold increase in risk of progressing to cancer, risk of progressing to blood cancer, sorry. And so we next decided to replicate this analysis for the other types of cancers that we had captured, so breast, prostate, and pancreatic cancer. And we find that we're still able to capture an increased risk of progressing to cancer, albeit more modest, in individuals who are harboring at least one somatic structural variant. So given the link between these large mutations and cancers, we became quite curious about the functional implications of somatic structural variation, or how are we getting from that DNA change to disease? And to investigate this, we decided to look at the downstream effects of those events, and specifically how structural variants might be impacting phenotypes such as gene expression. So what do I mean by this? Well, we might imagine in a purely neutral scenario that we could expect to see a reduction in gene expression following a deletion. We're losing DNA. It makes sense that we see that reduction in gene expression. And similarly, we might expect to see an increase in gene expression following a gain. We're gaining copies of DNA. We would expect to see that gene expression going up. And to see if this is in fact what's happening, we obtained overlapping genotyping array data and RNA sequencing data from around 647 individuals in the CanPath cohort. And quite simply just set out to perform a burden analysis to determine the impact of these structural variants on the transcriptome, so gene expression profiles across the population. So just to give you a very simple overview of how we did this, we rank ordered individuals by expression levels for each transcript, so one transcript at a time, and we start to capture the distribution of somatic structural variants across that spectrum of gene expression levels. And so what I'm showing you over here is the relationship between the cumulative distribution of somatic structural variants across gene expression percentiles for all genes which are impacted by a structural variant in the cohort. And what we find is that gene expression is not typically able to recover from those loss events, so loss and loss of heterozygosity. We start to see these really nice associations where we're capturing a greater burden of somatic structural variation at the extreme levels of gene expression, so very low and very high. And we think that this is likely due to the loss of a coding region or a key regulatory region, which might be controlling gene expression. And quite the opposite, we find that when individuals are gaining additional copies of DNA, we don't typically tend to see those same increases in gene expression, but rather we see that the greatest burden of gains occurs around the 50th percentile mark, or rather around the population average level of gene expression. And we think that this might be indicating the presence of stabilizing selection at the transcriptomic level, wherein even with an additional copy of DNA, average levels of gene expression tend to confer a higher fitness advantage to those cells rather than those extreme levels, which seems quite appropriate when you consider that we're largely looking at a cohort of healthy individuals here. So I'm just going to take a minute here to summarize a couple of the key takeaways from this latter part of the presentation. We're finding that a massive structural changes to our chromosomes in our blood are much more common than previously estimated, and they tend to be enriched at locations which have been previously found to be implicated in the early development of blood cancer. And we do in fact see that those variants are associated with an increased risk of progressing to cancer ultimately. But it's not that simple to figure out who is going to progress to cancer versus who is going to remain healthy. But we can start to tease that apart a little bit by integrating various forms of omics data, such as gene expression data, which help us to understand some of the downstream implications of those DNA level changes. And so what I'm hoping that you take away from these two stories that I've told you today is that there really is quite a diversity of selective pressures which are acting to shape how our blood evolves over time. And that we're thinking that if we can get a better understanding of the selective pressures and how they govern changes in our blood, we can really begin to understand when ARCH is benign versus when it really does require that therapeutic intervention, effectively letting us really capture the full potential of this blood phenotype as a biomarker for cancer risk. So with that, I'll end off by thanking many people, most importantly, the participants of the CanPath and EPIC cohorts, which really are the reason why we can do this type of research, as well as all of our funders and the CanPath team as a whole. Finally, a big thank you to Philip, my advisor, as well as our many collaborators and my lab as well. And with that, I'm happy to take any questions.
Wonderful. Thank you very much, Kim. That was a fantastic presentation and very rich. John, I'm going to again turn this over to you for the Q&A section. I don't see any questions yet popping up, so I would encourage our listeners to, oh, some questions are now starting to come up now. So to participate through the Q&A. So John, go ahead.
Thank you, Philip. And again, Kim, thank you. Congratulations on this, this tremendous work. A pragmatic and implications question from Stephen Nayrod. Kim, how would you intervene? Chemotherapy, question mark. What are the, what are the clinical implications for the future from your work?
That's a fantastic question. I mean, I think if I were to, sort of zoom out before even getting to the intervening aspect of it, I think that what we're really road mapping here is that there is a state where our cells are very, very healthy, in which they're optimized to die, right? If we're collecting mutations that are damaging, negative selection effectively removes those from populations and we keep our tissues healthy. Cancer is sort of really flipped that on its head, and those are instances where cells are really, they're thriving, even though they're capturing these mutations, they're forming those masses that we call a tumor. We're really looking at that area in between and helping to figure out what's going wrong. And where that becomes really important for treatments, I think, is it helps us figure out which clones to target. These patterns of negative selection that we're picking up on are almost these built-in tumor suppressive mechanisms where that selective advantage conferred by a driving mutation might be modulated by existing variability in our clones. So I think that that's going to become really important in both treatments, but also thinking about relapse.
Thank you. There are no other open questions in the chat yet. I think you've, maybe it's late in the hour, but also your work is very thorough, very thoughtful. Maybe a implication consistent with the first question, and that is, in terms of this platform, the work that you've done with CanPath and with EPIC, what distinguishes the ability to work with a prospective cohort like this versus more traditional clinical research, where it would be the identification of targets amongst cancer patients themselves? Any thoughts about the unique contributions of work from the prospective cohorts?
Yeah, that's a great question. I think it's speaking to, why we're all doing what we're doing with CanPath to a certain extent, is that I think that there's a huge amount of value to both clinical population cohorts, but also prospective cohorts, like what we're talking about with the European Investigation to Cancer Nutrition and CanPath. And I think that one of the key distinguishing aspects is something that both Nick and I have touched on a little bit today, is that we're not enrolling participants based on a particular disease outcome. We really are interested in collecting a snapshot of the population. And because of this, we're really moving the needle up on where we can start to understand disease. What is our starting point? And what we're getting at right now is that, looking at both Nick's work and the work that, Siggy Abelson has done with AML, and John Dick, and some of the work that we're talking about today, is we're not in a position where we're only able to study disease from the point of diagnosis on anymore. We're really able to start thinking about what is driving disease in a population from around 7 years prior to that diagnostic point right now, which I think is quite exciting.
Thank you. A question has come up from Jesse that how much genomic space do you need to cover for your evolutionary model predictions to be effective? For example, targeted panels versus whole genome sequence, also impact of sequencing depth. So can you comment on genomic space needed?
Yeah, absolutely. So I think Jesse's question is sort of in two parts. So the one is sort of the breadth of coverage. And the answer is not very much. So a lot of the data, that we're using now was drawn from a targeted sequencing approach, which really just looked at 261 genes. A benefit of our implementing this in a somatic context is that there's no recombination. So we have complete linkage at the cellular level. And because of that, even if we're just looking at a snapshot of the genome, we can really start to pick up on those evolutionary pressures which are acting genome-wide. The second question is more about the depth of coverage. And I will say that that's really the critical enabling factor of our ability to parse these evolutionary models. We had around 5,000x coverage per individual, which lets us detect extremely low frequency variance in the population, which is the signature of negative selection. And that's why we're able to start teasing out these different models. So, great question.
Thank you. Really, the last minute we have for today, Stephen Nayrot had commented that, congrats and that you have a fantastic resource. Maybe what I might comment on is that for all participants here today, this is our collective resource, CanPath, the Ontario Health Study, the regional cohorts. These are collective resources we would like, we built for all Canadian researchers to benefit from. And then my final comment is, Nick and Kim today, congratulations on your work. As introduced at the beginning, here's the work of two up-and-coming stars of the cancer research community in Canada. I think your PhD work looks fantastic and you've got great futures ahead. So again, further motivation for others across Canada to be working with these resources in the same way that you have. So with that, I thank you for your presentations today and to all the participants for joining us.
Thank you, Kim and Nick, and thanks to everyone. Fantastic presentations all to all. And I just will highlight as well something that came up during the presentations today. One of the projects that I'm very excited about is something that Kim is leading right now called the Canadian Cancer Study. It touches on a number of things that people are interested in with utilizing the CanPath resource. So please stay tuned to what we're hoping to develop with that. We think there are some exciting developments coming in the future. Thank you very much, everyone. Have a great rest of your afternoon.